Welcome, everyone. My name is Sarah Alger. I'm the director of the Russell Museum here at Mass General. Welcome to everyone here in the room and on Zoom. Before I introduce tonight's speaker, I'm just going to make a plug for our next lecture, which is going to be on Tuesday, June 10th. It's going to be called Courage and Resolve in Nation and Institution Building, the Influence of Doctors Joseph and John Warren on the Founding of MGH. They were the uncle and father, respectively, of John Collins Warren, whose bust you see over there, who was the co-founder of the hospital in 1811. It'll be delivered by Sam Foreman, who's both an MD and historian, who wrote a biography of Joseph Warren a number of years ago. Those of you who have been coming to our lectures for a long time might remember that he talked about the Warren dynasty a number of years ago here. So we're looking forward to welcoming him back. Just a housekeeping note, those of you on Zoom, just use the Q&A function to ask questions. And after Dr. Rosen is done, there'll be time for questions, which we'll take from both the room and from Zoom. So to introduce tonight's speaker, Dr. Rosen is director of the Athinoula A. Martinos Center for Biomedical Imaging here at Mass General. He's also the Lawrence Lamson Robbins Professor of Radiology at Harvard Medical School. He got his MD from Hahnemann Medical College in Philadelphia and his PhD from MIT. He completed an internship and residency at Mass General and research fellowships at Mass General and the Harvard-MIT Division of Health Sciences and Technology. Among just a few of his accolades, he's a member of the Institute of Medicine of the National Academies, the American Academy of Arts and Sciences, and the National Academy of Inventors. He's a pioneer in the field of functional neuroimaging. In the early 1990s, he oversaw development of the technique of functional magnetic resonance imaging, or fMRI, which I'm sure we'll be hearing a lot more about. And more recently, his work has focused on the integration of fMRI data with information from other types of imaging. And many of the tools that he has introduced are now used by research centers and hospitals around the world to study and evaluate patients with both neurological and psychological disorders. And with that, let's welcome Dr. Bruce Rosen.
Thank you very much for that nice invitation, and thank you all for showing up. It's great to see some old friends, some current friends, and hopefully some new friends as well. So thank you all for being here. As advertised, I will talk a little bit about brain imaging today. Although I have an MD and I'm a radiologist, so don't practice so much these days. My real heart is in engineering and physics, and so this will be a little bit more of a physics-forward kind of discussion. If we want to study the brain in mice or in zebrafish or fruit flies or any of the number of animal models, of course, we can take those brains, we can put them under microscopes, we can slice and dice them. But if we want to understand the human brain, of course, our tools are much more limited. In postmortem studies, of course, we can do certain things, but to study the living, working human brain, we need tools that are non-invasive and that have the properties that allow us to measure what the brain is doing, the work of the brain, whether it's its metabolism, its changes in blood flow, its electrical activities. And so a lot of work of mostly my colleagues at the Martinos Center have been focused on developing a whole series of new tools to be able to image the brain in ever more refined ways. And that's the topic of today's talk. So I'll talk about what some of the next generation of tools are, and especially put it in the context of what makes MGH a special hospital. We know it's a great place for healing. It's also a great place for engineering and physics. And some of you know that, but perhaps others don't, but you will by the end of this talk. And of course, as a physician, I am required to share with you disclosures, scientific boards and the like. I think the last line is the most important. Of course, as a physician, I need to announce if I'm talking about anything like a drug that's not FDA approved, I'll be talking almost exclusively about devices that are not FDA approved. These are devices in the development stage. One day we hope they will be, but most of them not quite yet. Okay, so there's a fundamental hypothesis that underlies this talk, and of course, much of what we do in science. And the quote from Freeman Dyson is, I think, a really nice way to capture it. Fundamentally, that new tools lead to new discoveries. So here he talked about the notion that new directions in science are launched in two different ways. One is by the development of new tools, and the other is by the development of new concepts. Now, he talks about concepts, and the effects of concept-driven revolutions is to explain old things in new ways. And think about Einstein and his general theory of relativity, which explained gravity in a new way from, say, Newtonian gravity, which is what we all learned in high school physics. But even more fundamental in his view was the effect of tool-driven revolutions, because they were the things that allowed us to discover new things that have yet to be explained. And that's why we're so keen on tools. So as an undergrad, I was an astronomy major. So when I think of examples of new tools leading to new discoveries, I think of this gentleman. All of you, I'm sure, recognize Galileo, who used this new tool at the time, the telescope, a pretty modest instrument, but yet just with that very modest instrument, which managed to magnify our view of the heavens, he really changed our understanding of humans' place in the universe. You know, from his observation of sunspots on the sun, he was able to measure that the sun was a rotating sphere and measure the period of that rotation. And the fact that it didn't rotate at the same rate everywhere, something nobody had any clue about. The craters on the moon, unobserved previously. The fact that the Milky Way was not just a band, glowing band, but it was actually millions and millions of independent stars, changed what we thought of that. And maybe most fundamentally, his very detailed observation of the 4 bright moons around Jupiter, the so-called Galilean moons these days, where he was able to show that there was a recapitulation of the model of our solar system, which had the sun at the center and the planets going around it, quite controversial at the time. And of course, Galileo himself got in trouble with the church for espousing it, but he was able to show around Jupiter the same thing, that there was this large central mass and the moons going around it, and the period of that rotation being dependent upon how far the moons were away. The whole story of our concept of where we were in the universe and putting the sun at the center and not the Earth itself, all was validated and verified based on his observation of these moons, something that could not have been done before a telescope. And of course, it's not just in physics that we see such things. Another name you're probably familiar with, Anton Leeuwenhoek, who, with, again, a very simple primitive instrument, really changed our concept of our physical world and our biological world, showing that there was just a whole realm of biological phenomena, creatures way too small for us to see with the human eye, and yet there they were, living and breathing and growing and doing all the things that they do, something that was just unobserved before, again, changing our concept of us and everything around us. So back to my theme of astronomy I love so much, and the title of the talk, of course, Great Observatories. When we think of observatories, we tend to think of telescopes. That's certainly what I thought about for many years. And up in space, there are these series of these great telescopes. Hubble, you've heard of, the Webb. There's a whole series of them. They all have different ways of looking at the natural world, which give us different views of what the heavens are doing. And by analogy, we like to develop tools to image inner space, the brain, looking at different phenomena within the brain. And I'll talk about this. So these are the great observatories. What are our great observatories in looking at inner space, at the space of the brain? Well, if that's the Hubble, that's the first of NASA's great observatories in space. I think everybody's heard about that. You know, I would say that the 7 Tesla MR, that's our Hubble telescope. That's really the thing that kind of changed our ability to look at the brain in its living way in unprecedented ways. So here at the Mass General, you might not think that we are engineers that build brand new instruments, but indeed, we did. And the very first in the world 7T scanner built on a clinical platform that was designed to study the human brain and human body was built here right at the Mass General Hospital's research facility in Charlestown, where the Martinos Center now lives. It had more than twice the magnetic field strength of any clinical scanner, really almost 4 times the strength of the routine scanners that most people would go in if they twisted their knee or they had headaches and the doctors prescribed an MR for it. And I think most important for the MGH's role, and we are, after all, the MGH Museum, it was built right here by MGH engineers and MGH physicists, of course, with help from our industrial partnerships, in this case, our partnerships with Siemens. There are 2 of my colleagues, Doug Kelly and Larry Wald. You'll see his name appear more than once in this talk, a brilliant physicist. This was in his earliest days with us, and he's continued to do amazing work since then. In fact, just recently won the gold medal from the Society of Magnetic Resonance in Medicine for his pioneering work, this being amongst it. Well, the big magnet was part of it, but it wasn't the only part of it. There was lots of complicated technology that got built that allowed us to not only take finer resolution images, but also take them much faster. This, in fact, is a tool that you do take advantage of today when you go in to have an MR, because used to be your MR studies would take a half an hour, and now they take just a matter of a few minutes based in part on this technology, the so-called brain bucket. Again, one of Larry's great inventions. The question is, what could we see with these combination of tools, the big magnets, these large, complicated arrays that we would put around the brain that we couldn't see before? Well, let's just take a simple example. On the image on the left is a typical image that we would take at our conventional clinical magnets, 1.5 Tesla. Tesla is just a measure of magnetic field. This is a patient with epilepsy. Of course, unfortunately for this patient, the epilepsy was not well controlled by medication, so they were imaging him to see if they might find the source of the lesion. And from that source, of course, they might have the ability to say, take it out, operate on him, and cure this patient. But when they looked at the conventional MR, they saw nothing. No target to look at, no chance to remove the lesion because they couldn't see it, and no chance to cure the patient. On the other hand, look at the 7T image, and I'll blow it up for you. There very clearly was a so-called cortical migration dysplasia. It's an abnormality of the brain that formed during development that was leading to his epileptic seizures. And now with the identification of a clear lesion, this subject suddenly becomes a good surgical candidate. And in fact, the fact that we tend to see these very small lesions is actually the best news, because these are the easiest lesions to remove and the ones with the most successful outcomes. And we've actually seen now in about half the patients that come to us with normal MRs, when we put them in the high field scanner, we're able to actually see something that then becomes a surgical target. These are patients that can go from a lifetime of poorly controlled seizures. They can't drive, just even, walking down the street can be dangerous for them if they suddenly have a seizure and fall in the middle of the street. Now they can lead normal lives. Now, of course, part of the challenge as a radiologist and a diagnostician, you need to know where to cut, and you also need to know when not to cut. So this is an example of that. Again, another patient with epilepsy. And sure enough, when we did the image, in this case at 3 Tesla, you can see a small lesion, and you might say, great, the source of this patient's epilepsy, we should cut this out. This patient will be cured. When you image it at 7T, notice the detail, these tiny black structures. These are blood vessels, so-called caput medusa type lesion. This lesion is actually one that's very dangerous to remove. It could actually bleed and lead the patient to be in worse condition after the surgery than beforehand. Something that was not at all apparent at the lower fields, and now we have a much better characterization of it, almost a pathologic characterization of it, but without ever having to put the patient through a risky surgical procedure. So not only do we see things that we want to resect, but we also have the ability to know when we shouldn't. This tool is not just used for studying epilepsy. Another one of the indications for this high field study is in the study of multiple sclerosis. Now, those of you who are knowledgeable about the history of MRI know that the ability to see the lesions in the brain with MRI in multiple sclerosis was actually probably the first killer act for MRI. It's because the CT scans at the time just weren't able to see it. The challenge was that it saw very clearly the lesions in the white matter. In fact, when I was in medical school, I thought of MS, it was a disease of the white matter. Those are the connections in the brain. But it turns out, if you actually look at patients, especially assess their, say, cognitive behavior, et cetera, that the relationship between the lesions in the white matter and their behavior, it's not very good. There's a relationship, that's just not a very strong one. There was something that was missing. Now, the pathologists actually had a pretty good guess to what that was, because when they would have the brain in a patient, say, after they passed away, and they would be able to do their pathologic studies, they could see lesions not just in the white matter, but also in the gray matter. But these lesions were very small and they were not very conspicuous. You would need a pathologist and a microscope to be able to see them. Sure enough, our colleague at the Martinos Center, Caterina Minero, was able to show that with the 7T MR, and of course the help of her physics colleagues like Thomas Benner, that she was not only able to see these small lesions in the gray matter of the brain, but actually characterize them just the way the pathologist did. Did they come down from the surface, the so-called subpial lesions, underneath the peel layer of the brain? Did they come up from the white matter, the glucocortical lesions? This is exactly how pathologists characterize them, and she could characterize them exactly the same way, but while the patient was alive and in the magnet. And of course, what she was then able to show, for example, in this case that she acquired, that in this particular patient, for example, who had significant neurological and cognitive disabilities, but only very small white matter lesions, something that couldn't account for the degree of her deficits, that in fact, when you looked with our high field tool, we were able to kind of clearly see within the brain these scattered cortical lesions that just weren't present before. So now not only could we understand her disease, but we now have a biomarker that we could use to see whether treatments targeted to that disease worked or not. And there are a series of studies today that are now looking at the different medications to try to see which ones work best for these cortical lesions. It would be impossible to evaluate if you couldn't actually see them, and now we can. So as advertised, I do have some historic interest in this field, the area of functional magnetic resonance imaging, or fMRI. It's actually my colleague, Jack Beliveau, who first published this picture, the one on the left, in Science magazine, which was the first image of using MRI, not just to see the anatomy of the brain, like in the examples I showed you previously, but to see the function of the brain, what part of the brain was working. In this case, you can see that kind of colored spot in the back of the brain right there. I guess that's showing up. That's the visual cortex of the brain. That's the part of the brain that's responsible for processing visual information. And you might gather, and you'd be correct, that this patient was actually seeing bright, flashing checkerboards, a bright visual stimulus that we were turning on and off, and we were able to see this. And of course, the amazing thing about this is not only could we see the functioning part of the brain, but we can see it connected to all the rest of the brain. In other words, we can combine the anatomy of the brain and the function of the brain with one tool, one image. And that had never been done before. In fact, this quite got some pretty good circulation. There it is, I think, on the South Pole. Clever, good luck on our part that he happened to be grabbing that journal. But where has that technology gone since then? And that wasn't done at 7T. What has 7 Tesla allowed us to do? Well, let's kind of take it a few years forward to the work of my colleague, John Palomini, one of our longstanding scientists, sadly escaped to MGH West at Stanford, as a few of our other colleagues have snuck out there. So an interesting thing about the way the brain is organized, its visual system, is if you look at an object and that kind of projects an image on your retina, the way a lens and a camera projects the image on what used to be film and now is your CCD devices, that projects right back to your brain, and there's kind of like a one-to-one image of what you're seeing in the back of your retina with what you're seeing in the back of your brain. And so John went to do a little experiment. He said, if I show a subject the letter M, what do I see in the back of the brain? You would expect that you would see this M. It turns out there's a little bit of geometry that you need to understand. While there's a kind of a direct mapping, there's a little bit of a geometric distortion. Through his work with Eric Schwartz, a neuroscientist at Boston University, he understood this mapping. So that's why he showed the subject this kind of slightly distorted M, hypothesizing that he would see this undistorted M in the back of the brain. And lo and behold, when he did the experiment, that's what you saw. Now you notice the first image, when it goes back, is the brain kind of crumbled up. The brain has these convoluted sheets. You've all seen what a brain looks like, kind of a piece of paper crumbled up. But if you take the paper out and flatten it, there's the M. Oh yeah, and for some reason, yeah, there we go. I just wanted to show it. So this is, there's a bunch of things that were required to make this work. Of course, we needed high resolution imaging. We needed the subject to be behaving themselves. We needed to understand how this brain all worked. And we needed the computational tools to take that crumpled sheet of paper, the raisin, and plump it back into the grape. That was what our colleague Anders Dale and Bruce Fischel did, this so-called cortical mapping. But put all those computational and technical tools together, and you could literally see what somebody was seeing. So we had a grant at the time. We thought we would try to spell this out on the back of somebody's brain just to show how brilliant and clever we were. Well, we got this far, but not a bad start. But in any case, it's very interesting because when you think about it some more, that primary visual cortex not only activates when you're looking, say, at this scene here, because your brain is also showing MGH in the back of it. But when you imagine, if you were to shut your eyes and imagine that MGH, your brain also lights up there. And when you're dreaming, that part of your brain lights up. So if we image your brain, could we see what you're thinking about and dreaming about? There are people that are actually doing these experiments now, and the short answer is maybe a scary one, but probably yes. We'll just move on from that thought. Okay, now modern neuroscience, as I mentioned, if you're looking at fruit flies or mice or something like that, we tend to think of modern neuroscience kind of from the bottom up, where we look at the synapses, the individual connections between parts of nerve cells, and then the nerve cells themselves, the neurons, and then the local circuits, how individual neurons connect to each other. And of course, there's amazing stuff that we can do. Our colleague Beth Hillman, a colleague of several of ours, showing these beautiful optical studies. Jeff Lichtman at Harvard doing detailed electron microscopy of individual synapses. Really amazing things you could do in animals at this very detailed scale. But the kind of human neuroscience that we think about is really kind of a top-down phenomenon, right? We can image the whole brain. We go down to kind of distributed networks, things like the visual system, memory systems, memory networks, a motor system, motor networks, down to detailed system maps, like those maps of the visual cortex that I was showing a few minutes ago. And of course, colleagues have been working on tools to be able to do this. The question is, where do they meet? What's the connection, the scale that kind of allows us to connect the dots between what the basic neuroscientists are doing and what the human neuroscientists are doing? Interestingly, they're almost like separate communities, like our stalactites and stalagmites here. If you go to the Society of Neuroscience meeting, there are 35,000 neuroscientists there. And, half of them are doing mice and fruit flies, and the other half are doing humans, and they go to separate sessions and they rarely talk to each other. But we think interesting things happen at that meeting point. And there's interesting questions. What are the properties of the brain cells and the networks that connect these scales? And that connection, that scale, is what we call the mesoscale. It's this middle scale between micro scale and macro scale, meso in between. And there are many interesting questions in basic neuroscience. The challenge is we need tools that can image at the mesoscale. And especially if we want to understand mesoscale in humans, we need tools that can image the mesoscale in humans. And so again, this is where these high field tools come to the fore. So many, many years ago, my colleague Roger Tutel took what is considered an iconic image That's this one on the left, which shows what are called cortical columns. The fact that at the next scale beneath, say, the map of the primary visual cortex, the brain is organized around these tiny structures, just about a millimeter or two, sometimes even less in size, that kind of interdigitate in this complicated way. He managed to do it very clever way. In this case, he did the flattening physically, but this was done, in a postmortem animal study, nothing that we could do in our graduate students. But these days, we can do the studies in our graduate students. And in fact, my colleague, Shaheen Nazar, actually was doing that and now is able to see these same fine detailed cortical structures, cortical column structures, mesoscale structures in the human brain using these functional imaging tools. So taking John Palomini's work, even the next step to the next level down in the scale. So you might say, well, okay, do I care about that? Well, it turns out that we probably should care about it. So for example, again, Shaheen Nazar has been looking at not just the normal organization of these mesoscale structures, but the abnormal structures. He's been studying patients with amblyopia or lazy eye. And it turns out the eye is not lazy, it's the brain that's lazy. And what he was able to show is that this notion of these interdigitated areas in the visual cortex, the blue, the green, the red, all subserving different functions. One that allows the brain to see stereo vision, depth vision, one to allow it to see motion, one to allow it to see color. Each one of these columns subserves a distinct function. But when he looked at somebody with amblyopia who didn't have stereo vision, didn't have this depth vision, sure enough, that part of the brain just wasn't there, and the brain had remapped, had reorganized around that. And so it has the motion and color areas and no stereo areas. And interestingly enough, now when people are proposing methods to try to improve this in the brain, now at least we have a tool to see whether they're actually modifying the brain the way we think we are, or can develop new techniques with a direct marker to see, is the brain reorganizing the way we think it is and should. And it's not just the visual cortex. I've shown a lot of that, but this is my colleagues, Roger Tutel. You saw that canonical image. He's still very active doing now. He switched from his work in non-human primates to work in humans, working with Daphne Holt, who is a psychiatrist who is very interested in this notion of personal space, and specifically in the setting this was during COVID when she first came up with the notion for the studies, where of course everybody's notion of personal space had changed quite dramatically, like 6 feet, right? But that wasn't the case before. And it turns out that abnormalities in the assessment of personal space are quite common in a variety of psychiatric conditions, like schizophrenia, like autism, and depression. Your sense of personal space changes. So the question is, well, where do these notions come from? What part of the brain subserves this? And she was actually able to show, with Roger's help and the rest of their colleagues, again, Shaheen Nazar, that part of the brain, the parietal cortex, a more forward part of the brain, that is actually responsible for this notion of personal space and the notion of motion towards that. Are you coming towards me, or are you withdrawing? Are you near, or are you far away? And again, there was this kind of interdigitated columnar structure that was never before visualized in a human, and it's directly related to kind of a very human phenomena of personal space. And again, now we have the ability to study this in a way that we never really had before. And it has important implications for studying a variety of psychiatric pathologies. Finally, I'll talk about another thing that we can do with this very high resolution and high sensitivity device, and that's look at time signals, very rapid time signals in the brain. So my colleague Laura Lewis was able to show that moving to 17 and even beyond, that she was actually able to see oscillations in these signals up to frequencies as to up to a hertz, changing rapidly, essentially on and off in a second. And you might say, well, why do I care about that time frame? Well, it turns out during sleep, for example, we have what are called slow waves in the brain that occur, and they occur right around that 0.75 to 1 hertz time frame. We didn't see that. In fact, you can look at these pictures in detail and see why we didn't. Notice at slower frequencies, the amplitude changes are like 0.5%. You might think that's small, but look at these changes at 1 hertz. They're 0.01, 100th of 1%. very small signal changes, invisible without the sensitivity of our new tools. But now suddenly we were able to see these slow wave oscillations in the brain. And you might say, well, why do I care what's happening in the brain when I'm sleeping? I'm asleep. Well, it turns out lots of interesting things happen during sleep. We knew, for example, that there were electrical changes, these slow wave electrical signals in the brain. And some people had hypothesized that during sleep, perhaps one of the functions of sleep was to remove waste from the brain, but nobody really had a mechanism that linked those two that allowed us to measure it. So Laura Lewis, a colleague of ours from MIT and also a member of our center, working with her colleagues, actually showed that synchronous with this electrical activity were changes in the hemodynamics and the function of the brain. And those changes in the blood flow of the brain, the hemodynamics, actually led to pulsations in the fluids in the brain. That's what's shown in blue. So the red is the blood signal. The blood is kind of coming and going in synchrony with those electrical signals. And it kind of in phase with that, or counter phase with that, you can see this pulsating blue signal, which is actually motion of the fluids in the brain. And so it actually is kind of one of the first indications of the fact that when we're sleeping, this coordinated electrical activity leads to coordinated changes in our blood flow and blood volume, which then leads to changes in the fluids. And essentially the brain is kind of pulsating, physically pulsating, and pushing the fluid around in and out, which is helping to remove the waste, so we think. And in fact, my colleague Zhejiang Ding is now, Zhejiang Dong, excuse me, is now using advanced MR techniques to actually measure the flow of these fluids in the brain in real time and 4-dimension. And of course, this is a topic of great importance because people think that in diseases like Alzheimer's, that there is a diminution in our ability to remove waste. Now we have a way to actually see if that's true or not, and potentially look at ways to mitigate the harmful effects of not getting that waste moving around. Well, if the Hubble is our kind of general purpose tool, the way to 7T, it's not their only tool in our arsenal. The Chandra X-ray telescope, X-rays are of course high energy objects, and Chandra was really designed as a unique instrument to look at high energy events. What's our kind of high energy event in MR? Well, I'm going to say it's our connectome scanner, and I'll describe this to you in a second. So a connectome is a map of how the brain is wired together. It turns out to measure connectivity in the brain, we need to goose up not this magnetic field strength. We don't need a bigger magnet, but we need another part of our system to be goosed up. If you've ever been in an MR, you've probably heard that knocking sound. That's something called the gradient, and that does the spatial encoding that forms the image. Well, it turns out that that is the key technical feature that is required in order to make measurements of how the brain is wired together. So our colleagues set out to say, well, if we really wanted to build a system that was dedicated just to see the wiring in the brain, how would it be different? Now, one thing is we would need to build much more powerful gradients. So that led to this so-called connectome scanner. You can see it's got 7 times the gradient strength, more encoding speed, almost a 10 times increase in its sensitivity, just to give you a sense for the energy required here. So there's our connectome. It's about 22 megawatts of power going through here. And that's about the same as a Los Angeles class nuclear submarine. So we're talking about a lot of power. Those of you who had maybe been paying attention to the indirect cost controversy, the government wants to reduce our indirect costs. Of course, now our investigators have a better understanding that that's not really a good thing for science. But for many years, we thought, well, indirect costs, why do we care about them? Well, actually here we care about them because the indirect costs pay our electric bills. So we're very happy for those indirect costs, and we certainly need them for lots of reasons. So these are the kind of pictures that you can take with that scanner, images of the connections of the brain. And these pictures have almost become, kind of ubiquitous in our culture. When you think of the brain, you've probably all seen pictures like this. These are amazing pictures that my colleague Van Wedeen, who was actually the first to do imaging of the tracts of the brain, this kind of tractography using magnetic resonance imaging, and is a computational visualization, graphic arts computer scientist, Rupeng Wang. They took beautiful pictures. They get on the cover of great important magazines. They get on the cover of music LPs, right? So these have now become a ubiquitous part of our culture. But it's not just beautiful pictures and great cover art, but there's some really pretty spectacular science that's come from this. And I'll just give like one example. And that's if you look at, say, this picture and now look at it in detail, I'm going to just blow up a section of it. You notice something really dramatic, that these connections of the white matter aren't just kind of like random bowls of spaghetti wires kind of going everywhere the way if you would open up an old computer, the wires would be going in every which way. But there's like a regular pattern to how these white matter fibers are organized. They seem to be going in one direction, largely in perpendicular directions, and then even in the third direction, kind of the avenues and the streets and elevators, if you will. Van Wedeen, the guy who made this discovery, is a New Yorker, so that's the way he thinks about things, just like the layout of Manhattan. And he called this grid diagram. It was very controversial when it came out. It still is a hypothesis and an idea that's evolving, but it was a really notable observation that actually turns out to have at least some element of real biological basis. His colleague at Boston University, Doug Racine, actually did studies in animals to validate this and showed if you inject some part distant, that if you look distant from that part of the brain, sure enough, you see that same grid, the same pattern of some fibers going this way, some fibers going this way, and you would see fibers going in the perpendicular direction too. So why is it, and what's important about this grid pathway? Well, we're still really trying to understand that, but it does kind of change the way we think about how the brain is wired, from a picture that maybe looks like this, where everything kind of just going in its own direction, to maybe something that looks a little bit more like this. And what are the implications of this for how the brain develops, how the brain can be plastic in the face of disorders like strokes, how the brain may have evolved, where we see evidence of these grids in very primitive creatures. All super interesting questions, but not the only kinds of questions that we can answer with this amazing tool. Another one of our colleagues, a wonderful neurologist at the Mass General, Brian Edlow, is interested in disorders of consciousness, so things like acute coma. Back when he was a medical student, he had the opportunity to work with this young fellow, 20-year-old college student, riding his bike, got hit and run accident. He's in a coma. Here he is 14 days later, still unresponsive. And the question that parents, of course, ask is, what's his prospect for recovery? Based on the fact that 14 days later, he's still unresponsive, the answer was not very good. But parents, didn't give up hope. Here's the MRI taken at 7 days, and all of you probably see this. Turned out this is like nothing. This is not his problem. Let's see if I can get my cursor. This little guy, that's the problem, because that's in one of the key parts of the brainstem that involves arousal. There it is. Always putting an arrow where you want people to look. Well, amazingly enough, fast forward several years, there he is on his bike, actually literally rode his bike cross country. You might argue maybe something about his brain that didn't quite recover, because he could question whether it was good sense to ride across country again. But he did so. He was doing great. There he is at our connectome scanner. And there's my colleague, Brian Edlow, with his young patient that he looked up several years after the events. So the question in his case is, could we understand how it was that he was able to recover? So sure enough, they put him in the connectome. There was that day 7 image. And then here we took another picture, this one in our 7T scanner, so we could get a much more precise view. And we could outline the fact that we saw this kind of area in blue, the PPN, that stands for the pedunculopontine nucleus, a fancy thing. The red thing is the hemorrhage. But you notice that fortunately for him, that hemorrhage kind of transected the pontine nucleus. But many of the fibers were still intact. So by good fortune, it had been the other direction, he might have knocked off more of his fibers and he might not have recovered at all. But in this case, many of those fibers were intact. And so in retrospect, we could see, now we understand why he could recover. The question now that Brian is asking when he's seeing patients who are in their comas, can we predict who can recover or not based on what we see now? And he's now doing the studies to be able to see, will the function of the brain, will the connections in the brain give us the ability to see who can wake up and who won't. And he's making great progress on that count. Well, we never quit. Connectome 1, 22 megawatts was good, but clearly we wanted more. And so my colleague, Susie Huang, decided, to let's go to Connectome 2.0, the next scale of this device. And in fact, why? Well, if we really want to probe kind of nerve axons, micron scale structures, we needed to push the gradients even harder. We wanted to work with colleagues like Jeff Lichtman, who could help us validate what we were seeing. And this would really allow us to kind of learn the detailed nature of the connections between structure and connectivity and function in brain development and healthy aging and various disease states. So again, we wanted to start with that gradient coil. We built the next generation of coil. There's the scanner. Looks pretty much like a normal scanner, only this one is only really big enough to be able to put your head in. But what's really important here is these numbers, which shows that it has almost 4 times the oomph as the Connectome 1.0. So not quite an order of magnitude, but we're gaining on it. Another really important improvement. So one of the things that we discovered, though, as we really pushed these gradient systems harder, is that we had I wouldn't say fully unanticipated, but an unwelcome effect. And that's these very rapid changing magnetic fields would actually stimulate peripheral nerve cells and people would feel it. And then, of course, if that sensation got strong enough, it would feel painful. And you might say, well, what good is that? You built this very powerful system and you can't use it because the patients want to climb out of the magnet. So it was a real problem. We needed to find a way to get the maximum performance, but yet not generate these peripheral nerve stimulation problems. So here's our hero, Larry Wald. We talked about him earlier, still working. Lost a little bit of hair since in the 2 pictures, but the brain's still fully functional. And a wonderful young colleague of his, a postdoctoral fellow, Matthias Davids. And what they found is that if they built very detailed models of where the nerves, they actually looked at models that had thousands of nerves in the body and put in all the properties of those nerves, and then put in the properties of the gradients and where those magnetic fields would be generated, that they could actually predict not only where the stimulations would occur, but they could show how you could modify those gradients in order to still get the scanner to work just as well, but not produce those effects. And so they're actually able to recover more than twice the performance of the system just by being able to make minor modifications to this gradient system to allow the system to really work at its maximum. Detailed biophysics, but a very important contribution to studying the underlying biology. And interestingly enough, another colleague of Larry's, Valerie Klein, and Bastien Guerre, actually kind of now reverse this situation. If we know how to model how to stimulate nerves inadvertently, could we flip that around and say, well, what if we wanted to stimulate the nerves, like in somebody with spinal damage? And sure enough, they're now in the process of designing systems that actually can dedicatedly stimulate nerves. And it turns out with magnetic fields, it looks like we can do a much better job than electrical stimulation, which has already been shown to be beneficial. So we're very excited about these prospects. Well, so here are the first brain diffusion images with the Connectome 2.0. Not the healthiest looking brain. Actually, it's because it's my brain. I too have lost a little something over the years. Little hair, a little bit of gray matter as well, but the images look pretty good. And indeed, even compared to this amazing Connectome 1.0, much more detail in terms of the connection and wiring of the brain. And our colleague who's sitting right here in the audience here, Anastasia Indicki, I didn't put this slide in knowing she was going to be here, but I'm sure glad I did. In any case, she's now using this tool and a variety of other very sophisticated tools to try to build an entire map of every axon in the human brain. An amazingly ambitious project, but one that really will provide us a roadmap for how the brain is wired and organized that we've never done before, using MRI, using tracer injections in animal models, using fancy optical techniques like polarimetric sensitive optical coherence tomography, labeling, micro CT, and electron microscopy. An amazing connection. Talk about bridging those scales. She's really literally doing it from the stalactite right through the stalagmite and all the points in between. Ambitious project, but a really amazing one. Well, okay, so even Hubble can occasionally get an upgrade. We actually took that lesson from those gradients that we built for the connectome. We said, well, maybe we can build an even better 7T scanner. And sure enough, we did. This impulse scanner was designed again by MGH engineers, Matthias Davids and Larry Wald, help from our colleague David Feinberg from Berkeley and our friends at Siemens who actually did the construction for us. And this system is just coming online at University of California, Berkeley. We have another one coming here to the Mass General very soon. The sky's the limit on what we're going to be able to see here, really just beginning to work here. Well, probably my time is getting short, so I'll try to work relatively quickly through this, just maybe the final telescope that I'll talk about. The Spitzer, of course, is an infrared telescope. An infrared spectrum really allows us to see molecular chemistry in the universe. So what's our kind of chemical equivalent? And our chemical equivalent is, in our tools in looking at the brain's chemistry, is positron emission tomography or PET scanning. But of course, never wanting to leave a technical challenge unattempted, we decided, well, you really still want the anatomy and the function of the MRI and the chemical sensitivity of PET. Why don't you do them together? So sure enough, my colleague Chipper and Katana came here. We were the first to have a human scale PET-MR system, and it combined the strengths of MRI, like the high resolution you've seen, the great physiology of, say, brain function. It's non-invasive, but it has its challenges as well. PET, on the other hand, has exquisite sensitivity to the chemicals and neurochemical processes in the brain, but its resolution is not as good. The question is, could you put the two together to get the best of both worlds? And the short answer is, yes, you can. And that's what we've been doing with this new tool, this PET-MR tool, which has allowed us to study all sorts of neurochemical processes in the brain. I'll just show, just two quick examples. One from the work of Marco Logio, who is studying brain inflammation in a variety of conditions like ALS and other neurologic diseases, but he was interested in brain inflammation during the COVID pandemic. And of course, one question was, well, is there inflammation in the brain during COVID itself? But he came to discover that in many of his uninfected volunteers, normal volunteers, and in fact, post-COVID, their brains were also showing evidence of increased inflammation, presumably from the stress associated with COVID. And so we now understand that there's a close connection between brain inflammation and brain stress. Based on these data, we can actually now look directly for these kind of effects. And then showing that similar biochemical changes were also seen in the MR spectra. So even in non-infective individuals, COVID was not really good for us. I think we all know that. And of course, we talked about functional MRI, where we can turn on the lights and see the brain turning on with our bold imaging or flow imaging. We could do the same thing looking, say, for changes in metabolism in the brain or chemistry in the brain. Here's a study where we looked at the brain's use of glucose and actually showed how it increases in the same way that the blood flow does. And ultimately, of course, we want to be able to do this to look for receptor functions in the brain. How is dopamine turning on when we're saying a rewarding task, or serotonin if we're interested in studying patients with depression? To do that latter task is very difficult, requires an even more sensitive device than the ones we were able to develop commercially. And so my colleague, Chipper and Katana, is now in the process of developing the next generation of PET-MR scanners They started with really a novel design, almost a spherical PET system that you put your head in, and then had, of course, to build all the rest of the components, including the MRI components, because his goal was to put this inside the 7T MR. So the highest resolution PET camera ever built, the highest sensitivity PET camera ever built, and put in our ultra high sensitivity MRI system. Really remarkable. He's got these pieces together. I'll just show the resolution, almost a factor of 2 better than any of the other PET cameras out there. And the sensitivity almost 10 times bigger. And why would we care? Well, that means we can reduce the dose by a factor of 10. We can image multiple times in a single individual, maybe image children, because the dose can now be, as low as the same radiation dose you might get with a cross-country flight. So we're really excited about having this extra sensitivity, and this we think will be the sensitivity we need to be able to look at neurochemical changes dynamically the same way we look at blood flow changes with fMRI, and that will be an amazing capability. Well, is that all we got? No, there's, of course, lots of other things we can talk about. Handheld MRI. You can do that, right? Clarissa Cooley is doing it. Magnetic particle imaging, another one of Larry Wald's amazing devices. We can put together ways to stimulate the brain, like transcranial magnetic stimulation, with ways to measure the magnetic fields in the brain, MEG, with MRIs, and put these all in one crazy setting. Portable imaging with optics. We were talking earlier about the wearables. We can now have these kind of wearables right in the neonatal intensive care unit to watch the function and the metabolism of the brain in real time. Lots of other fun stuff that we could talk about. But next talk. So if the 7T is our Hubble, then what's our Webb? What's the next big thing? Well, so here's a picture of the brain with resolution of about 100 microns, a tenth of a millimeter. Right? Pretty amazing detail in the picture. So that's the good news. We did this with our 7T imaging. Fantastic. The bad news is that this was the brain in a bucket. This was a postmortem brain, and it took 24 hours to acquire. So amazing, but not really practical for most of our clinical studies. So, how are we going to take these kind of pictures? The machine is capable of it. How are we going to do this? Well, we just got to get more oomph. So, if we went from 1.5 to 3 and from 3 to 7, 7 seemed like 14 is the next step. And sure enough, you get, more than twice the performance, more than twice the resolution, and well more than twice the cost, I suspect. It's going to be an amazing adventure. Andrew Webb, no connection to the Webb telescope, I don't believe, but there it was. Now, in fact, somebody's actually building this device. So that last diagram you saw, that was theory. The group at Donders Institute in the Netherlands is actually beginning to build this now. So if we're going to build one here at the MGH, we better get cracking. So anybody know anyone with $50 million to spare? It's about what it's going to cost. Please give me a call. But somebody's going to do this, and I think it's going to be pretty amazing. So I'm going to close with one final point to make. We talked a lot about tool building and how important it is, but of course, it's not just the tool building that's important. So this guy, you probably have never heard of, right? George Ellery Hale. In fact, has anybody who hasn't seen this talk before ever heard of this guy? There we go. Thank you. There's an educated man. Right. Well, as well, you all should know him because he is the greatest telescope maker, really of all time, certainly in the 20th century, maybe equivalent to, Newton with his first reflecting Newtonian telescope. Right. He built the largest refracting telescope in the world, the 40-inch at Yerkes, the 100-inch Mount Wilson telescope, the 200-inch at Palomar. These are the classic telescopes of our generation. Thank you very much. But most of us haven't heard of it. But this guy, you've all heard, right? So why was the space telescope named after Edwin Hubble and not George Ellery Hale? Well, first, why is it named after Hubble? I mean, we've all heard his name, but you may not know why it's famous. So what he did was he took these telescopes and he looked at a class of stars called Cepheid variables, which had this interesting property. They were variable stars. They kind of got bright and they got dim. They got bright, they got dim. And what others had shown previously, is that period, how long it took them to go from bright to dim, actually was a reflection of how bright they were, like in absolute terms. Not how bright in the sky, because that would depend on how far away they were. But if you had them all lined up, all right next to each other, the brighter ones, now I'm embarrassed to say, I think they oscillated faster, but I got a 50-50 shot of getting this right. Could have been slower, one or the other. But the bottom line is that period, whatever, would tell you the luminosity of the star, like in absolute sense. So that was great if you did it in nearby stars, but you needed a big telescope if you were going to see them from far away. And in particular, if you wanted to see them in other galaxies, which people didn't even realize were other things like our Milky Way, but much further away. They knew this by Hubble's time. And what he did was he actually looked with these huge telescopes and he looked at the Cepheid variables, not in our Milky Way, not in our galaxy, but in distant galaxies. And he showed this relationship, that there was a relationship between how far away the galaxy was, which he knew because he knew the luminosity of the star, because he knew the periods, and how fast they were going away. You could measure that with the Doppler effect. So what he showed was the further away it was, the further it was going away from us. In other words, he showed the universe was expanding. Again, a fundamental observation that changed our fundamental understanding. Of course, you unwind that, you wind backwards in time and you get the Big Bang, and all of modern cosmology come from that observation of the fact that the universe was expanding. And, quite an important discovery. So my point here is that, yes, building the tools is important, but using them is important, right? And sometimes the users get, get the greatest fame, but don't forget the guys that built the telescopes. They could never have done it without the guys that use them. And the same is true for our tools. You're going to see over the next few years amazing discoveries from Mass General investigators studying all sorts of diseases of the brain. Don't forget the Larry Walds and Anastasi and Dickies of the world for the amazing things that they built that have allowed those to understand human disease. So these are my thanks to all of my colleagues who so generously lent me their slides. And thank you all for your very kind attention. I know it's getting late, but I'm happy for a few questions if you got them.
Amazing thought. I cannot believe you summarized decades worth in this one. Fantastic. I have a question on resolution, but not the resolution that we were talking in millimeters, but rather in time. We know the brain changes over time, either whether you learn a task or some other way. What is the shortest in time that you measured and seen the brain change? Any specific area of the brain?
Yeah. Well, essentially, as quick as we can image, we can see changes in some things that we look at. So, for example, blood flow, that changes very quickly. When those lights turn on, your blood flow changes within a few seconds, and we can easily track that. To my mind, an even more interesting question is, how about things like plastic changes in the brain, like in learning? Can we see those changes? And it turns out there's some evidence now that those changes also occur very, very quickly that we can see. So, for example, there's a group that's done, learning tasks where they had to learn a procedure, like tap your fingers in a certain pattern. And using these same diffusion tools, we actually can see changes in the diffusion in the parts of the brain that we know are involved in learning the tool, because we see those parts light up when we do the functional studies, and they change within minutes. So something is happening to the microstructure of the brain within minutes that then subserves this kind of procedural learning task. Nobody today knows, like, what are the microstructural changes that are occurring on those short time frames, right? We're not necessarily sprouting new neurons in that time frame, but something is going on. So almost at any time period we look at, we see something interesting. And at the opposite scale, it's actually harder to measure changes over a very long period very accurately because things like the machines drift. If you wait long enough, then you get new machines. And how do you compare a measure you made with one machine and another? But that's also a challenge that people are taking on now, for example, to try to measure changes, say, just even something simple like the thickness of the cortex, but do it very, very accurately. So we can see in patients, say, that we're suspicious of having dementia, but we don't know. Can we see, say, with a follow-up visit and just a few months later, can we see very subtle changes? And again, if you're very careful about your engineering and your techniques, it looks like we can see changes in a matter of months that previously would take years to be able to see. So almost any temporal scale is interesting. And you just have to have the right set of tools to kind of match to the thing that you want to measure.
Other questions from the room?
Well, I know that means one of two things.
Thank you, Bruce. You started your talk mentioning the revolution in technology and then the revolution in concepts and how the change will be explained. And since we're in the museum, I would ask your thoughts. If you were to go back before fMRI was invented and everything we've seen today with these new tools, what would have been the most surprising thing if you were to go back to the days of those first fMRI images with everything you've learned today and then say, wow, this has fundamentally changed everything that has come before?
Oh, that's a really good question. How can I rephrase it to say, what have we learned from our functional imaging studies that's really kind of changed our concept of how human brains work? Is that a fair way to get at it? I think one of the important things kind of follows up what I was just talking about it, and that's the tremendous plastic potential of the brain, the ability of the brain to continue all through our lifespans to change in the face of learning new things, adapting to conditions, adapting to disease perturbations, which is a good thing. The older I get, the more I appreciate the fact that our brains do have this amazing plastic potential. That's something that, I think we understood at some level, but now we can actually see those changes and appreciate them. Each of the detailed systems, people that study memory now have a much better understanding of the kind of the architecture of memory. People that study the visual system now can look at columnar level structures. So within each kind of every neuroscientist specific domain, they see changes. But I think more fundamentally, to me, the most interesting thing is just how our brains are able to adapt and change and relate that kind of physical structural changes to the cognitive and behavioral changes that we can see.
I'll ask a question if I may, and you're welcome to throw me out the window for my asking. I wondered what your thoughts are about AI for both your center's work, and if you have any general thoughts you can share, given how much you know about the human brain and human intelligence.
The first
There's
part,
a window right here.
yeah, no. It's a great question. The first one is easy, which is AI is now a ubiquitous tool, the same way that basic mathematics was the ubiquitous tool, when I was in school. There's almost nothing that we do that, doesn't use AI in some sense. It's used in how we produce the images, how we so-called reconstruct them, how we analyze them, what we look for in our image. All those tools are now either AI-based or AI-informed. So, it's completely embedded itself. And that's been a remarkable change, right? I mean, that was 5 years in coming, and it's just changed the way we do science. So that's amazing. What do we know about, what do we think about, like generalized artificial intelligence and how that relates to what we know about the human brain? I'm probably not the right person to answer that question. I mean, people certainly are thinking about building, kind of biomimetic intelligences. And I guess the question I've got is that the way forward? I mean, that's an interesting exercise in trying to understand how the brain itself works and then trying to use modern computational tools to mimic that. That's tricky if you don't really know how the brain works. And so we've got a little ways to go, I think, before we're quite ready to do that. But then the other question is, well, is that really the secret to that generalized intelligence, right? Does it have to work like a human brain to act like a human brain? And if you believe that we're making progress in that direction with the AI tools that we have, the answer may not be yes, right? I mean, we talk about neural nets, and so yeah, there's some kind of very loose homology to how brains are organized, but it's quite loose, at least in my understanding. And it's not so clear that the tools that modern computational scientists are using to build these amazing, ChatGPT and all its successors are really fundamentally built on neurobiological understanding. I think for the most part, they're not. It's a good part of the story, but I think it's more story than reality. It's clever computer science. It's not necessarily adaptive neuroscience. And maybe that's why the AI tools aren't what they need to be. And we're missing some key ingredient that biology has learned that we haven't put into our machines yet. I'm guessing that won't be the case, but that's just a guess.
So much. Any last questions before we part? Yes.
No.
What's your prediction of the limits for MR? So will 14 Tesla be the upper, the last hurrah, or what's after that?
That's a really good question. It's taken us a long time to even get to like 11.7, close to 12T. 14T is going to be really hard. It took us, it's been 20 years since we've been playing at 7T, and it's only now kind of like hitting its stride. The biological effects also scale, and some of them, like, I showed with Larry in the PNS, I mean, you can work around, others are harder to work around. I'd like to go in a 14 T scanner myself before I set out to build a 20T scanner. see how that goes. I think it's not entirely clear, right? I mean, we put animals into much higher field scanners, but they can't really tell us how they feel and whether they're happy to be there or not. I don't know. 14T will be a great challenge, and I think it's going to be, a decade or two's worth of hard work to get that to work. And then maybe we'll be in a better position to guess. One thing is certainly true, in that the AI tools have made a big difference, and there are things that we can see at 7T with those tools that we couldn't see before, but then at 14T they'll work even better. So that's not a reason not to go higher. It's just to say that we can creep up in a number of directions. Come back in a few years. Build that.
That will invite you back in a few years if you're willing to come back. Thank you so much.
Thank you all for showing up on a